So tonight we're privileged enough to have Dr. Yi come in and give a presentation to us. So we normally refer to her as Eva.
Yes, call me Eva.
So you guys, when you ask questions, can refer to her as Eva if you want. Now, I've known Eva since about 2003, so about 7 years. And I guess she's had to put up with me since. But she's going to give us a talk over simple nitrogen oxides in human health, chemistry meets biology. So hopefully you guys notice the important words in there. Chemistry, right? And so, another nice thing about OU right now is, well, Eva's a research assistant professor at OU, working under Dr. Victor Otto. And they just got to move into a brand new research facility. So what was the cost on that? Is there?
I think it's 80 million.
80 million?
Yeah, 80 million.
So an $80 million facility. So they do good research here.
Yeah, I will show some pictures at the end of the talk.
Anyways.
So if you guys are interested in the future for the graduate school, definitely welcome to look at our OU chemistry department. Very nice facility and brand new building, very good. So if you want to stop by sometimes and before you graduate, you can contact Dr. Wickham or contact me. I will give a talk when you arrive at OU. It's very nice.
I've been there. It's a nice building.
Yeah, okay.
So,
yeah, we have more.
anyways, I'll turn it over to Eva here and let her talk to you. If you guys have questions, ask her at the end of her talk. And if you have any questions about the programs at OU, the chemistry program or buildings, you can ask her about that as well.
Yes.
All right. Thank you.
Thank you, Jason. So everybody can hear me? The voice is not enough. Okay. Okay, today I will talk about the simple nitrogen oxide in human health. It's like a big topic, but when you get it down, we'll narrow it down. And chemistry meets biology. Before that, I want to briefly introduce the global nitrogen cycle. So it's very important, we know, because you need the amino acid for the protein build up. Amino acid is a building block for the proteins. You need that, but you need nitrogen. So the nitrogen cycle is processed with nitrogen is converted into the ammonia acid. So they make proteins used by the photosynthetic plants. So the plants the plants here, they build the ammonia acid. Ammonia acid that will be used and then to make the proteins. Okay, so this transformation can be carried out by biologic or non-biologic processes. The biologic processes go to the diazoal choppers, so basically to use the nitrogen to make the ammonia. And another one, non-biologic, normally, so it's like a chemical way, it's called Haber-Bosch process. Basically use the nitrification to make the ammonium. So, but use a chemical way, it's used high pressure and high temperature. That is a non-biological way. So the important process in the nitrogen cycle, including the nitrogen fixation, and this is fixation, and the nitrification, and then go back to the denitrification. But human activities involved in the nitrogen cycle actually change the natural nitrogen cycle a lot. Why does that happen? Because we know the food, we need the food. So that's why before 200 years ago, without the good fertilizer, so the product of the plants and the crops is lower down. But since the fertilizer comes on, so it increased the food by increase the plants, crops, and then they will use food, fiber, and wood. So that is very good. So human activity involves the nitrogen cycle. But today I'll just mention about the small molecule, nitrogen oxide. What does that do? So the general outline is what and where is nitric oxide, and why is nitric oxide important, and how is produced. We call it We not call NO, we call NO. So it's how is NO produced in the biological systems. And heme, you will know later, nitride and NO, what are the relations? So where we can find nitric oxide? So at high temperature combustion devices, or maybe in the nature, so when the lightning, the electron sparks hit on the gas, so atmosphere, so you have a nitrogen. We all know the atmosphere is 79% is nitrogen, right? So when the lightning, so the high temperature and high voltage, it will form the nitric oxide trace. But since the oxygen around, nitrogen oxide will goes to the nitric dioxide, and then the reactions goes on. Also, the automobile exhaust contains NO, trace of the NO, and the industry to make some acid, they will form some trace of the nitric oxide. And also the upper atmosphere, so you have a supersonic jet. They use a nitrogen-containing fuel, so that will also give some NO. So in the living things, like bacteria, fungi, we convert nitride to NO. And the major reaction of the NO is converting back to the nitride, because NO is not stable. You either go to oxidize or reduce. So they use the nitride to convert to nitride by reacting with an oxygen. So that will, in the air, when you have a trace of a nitric oxide, that will be oxidized by the oxygen to forming the nitride small. And reactions with the non-metal and metals. I want to mention out some of the NO donors. So the ammonium nitride, maybe you may or may not know, in the 80s and the 70s, they're very popular in the club because they have a name called the poppers. I don't know whether you guys know that. I don't. So it's kind of make you relax, it's fun, and this is kind of what is chemical formula is that ammonium nitride. And this is nitrous oxide also is give a drugs for the NO donor to vasodilate, so it's biologically relevant. And if you notice that the NO attached to oxygen at the ammonium nitride, but this side is the NO attached to sulfur, sorry. And this NO is attached to iron, so basically they can have different NO donors. And in my alternate slide, I mentioned heme. What is heme? Heme basically is a porphyrin type, but mostly is iron containing. So here is iron, so Fe is a symbol for iron. So the porphyrin, so this is you can find about in most of cases. Your blood is red. Why? It contains hemoglobin. Hemoglobin contains heme. In muscle is red, myoglobin. It also contains heme. White plate is showing green color. It's not a hemoglobin, but it's chlorophyll. It's a kind of type of the heme, the porphyrin. And so the synthetic models have been studied for quite a while. They have a different metals instead of iron. They have a chromium, molybdenum, and magnesium, and ruthenium, and so on. But in the natural systems, you can find mostly it's iron. So you can find it in the NO synthesis, in cyclone P450, and the myoglobin, and the hemoglobin I just mentioned, and the nitrogen, some interesting protein I will mention a little bit, and the nitride reductase. So that is a heme protein is important, and they can react with NO. When the NO binding with the metal, we call the nitroso. M stands for metal. And with X means non-metal, so it's like sulfur, oxygen, we call it the nitroso. Okay, so give a little background. Why is NO important? We kind of a little further, we talk about Alfred Nobel. Everybody knows Nobel, right? Give the Nobel Prize. So once a year, it's very, the most famous prize. Every scientist's dream is get a Nobel Prize. So here's one, the name is come from him. So he was born in Stockholm in Sweden in 1833, and he invented dynamite. So that one of the most famous patent. he has. But he held like more than 350 patents. But dynamite is most famous one, and made him fortune. Besides the chemistry, he also is an engineer, innovator, so he's done a lot of work. So as I just mentioned that dynamite is the patent of one of his patent. So this is a major component of the dynamite. It's called nitroglycerin. If you see, this is kind of unique. If this is pure nitroglycerin, it gets oily, colorless liquid. But when you explode it with high temperature and combustion, you will get all gases. It's like 6 nitrogen and 12 carbon monoxide and water and its gas form, and 7 molar of oxygen. So it's 4 molar of the solid. You can get 35 molar of the gas. So that is very explosive. It's very dangerous. So this is a major form of the dynamite. What's the relation of the dynamite with NO? Later on, in 1890s, nitroglycerin recommended as a drug for Alfred Nobel. What is the component for the dynamite to treat the patient? So it's kind of ironic. In the later, that is October 25 in 1896, it was saying, isn't it ironic fact that I have been prescribed nitroglycerin to be taken internally? So he has taken the nitroglycerin, the dynamite, and put it under the tongue. And they call it trinitrin. Nobody wants to say it's nitroglycerin. Otherwise people won't take it. And so as not to scare the chemist and public. Why is that? And later on, after 100 years later, in 1998, the Nobel Assembly at the Karolinska Institute in Stockholm, in Sweden, so they has awarded Nobel Prize in Physiology or Medicine for 1998 to 3 scientists, Robert Foucault and Louis Ignoro and Frederick Murat, and for their discoveries concerning the nitric oxide as a signaling molecules and in the cardiovascular system. So NO is now known to be associated with many biological events like vasodilation, so that is cardiovascular systems, and memory transmissions like neurotransmissions, and also involved like DNA damage and feeding of some insects. What is it? So we now know the NO is potent, and it's called vasodilation. So why nitroglycerin is prescribed to Nobel? Because they can be treated as NO donor to cause vasodilation. Okay, so this is one of the Ignaro's experiment. What he did is, we know that what they call it, beginning is called indocerium derived relax factor, but nobody knows what is that. One of his experiments is that he mixed the hemoglobin, the reduced form, the red color, with the EDRF, the indocerium derived relax factor. They show the spectra from the brown one shift to green one. They repeat this one, but they use the NO, nitric oxide, and with the hemoglobin, he gets the same spectrum. So that means UV spectrum will be one of the tools to determine, oh, right now, of course, more experiments need to be done. This is just one of the examples. They say, okay, so EDRF is NO because they give the same spectrum. This is one of the experiments. And how is NO produced in the biology? So NO, nitric oxide, is produced every second in your body, everywhere, in the muscles and in the brain, in some of the cells for the immune system. So the enzyme that produces NO is refers to NO synthesis. And NO synthesis contains heme. So that means the heme is also important. What's happened, the reaction it catalyzes the terminal nitrogen at the end of the arginine, at the terminal of the arginine, in the presence of the NADPH, this is a kind of a cofactor with plus the oxygen that will give the NO and the citrulline as a product. So the NO is produced by the arginine in presence of the NADPH and oxygen. So I give examples of the NO, why it's important. You will see in the daily life. So NO with fireflies. You will see the fireflies give the light, right? Why that happen? Is that the light, to produce light, you need oxygen. But in some enzyme, so they can bond with oxygen to quench the oxygen, so you cannot give light. So the NO involved in these processes, the luminescent cells contains NO synthesis, basically produce the NO. NO and binding to the protein that also binds to the oxygen. So this way, NO inhibits the close by enzyme that binds the oxygen. If NO binds the protein, oxygen won't bind it. So oxygen is free for the nucleus and then give the light. So the more oxygen is available for the light produced, you will see. Actually, you see the pores, that is NO formation, NO bind the protein. So the oxygen combined with the luciferase and give the light. So it's very neat chemistry, but it involves NO. Another one example is, this is a Rhodinius prolixus. So this is a bed bug. So you will mostly find it in the Central and South American. But right now the trend is moved to the north. You can find them in Texas, Arizona. And this is, so this feed on, this bed bugs, they feed on the blood of the rodents or large mammals like humans. And a slow feeder. So you will see, and they poke around until a suitable wound is created. So they just poke them. And the funny thing is they tend to the people's face. So there is a name is called a kissing box. So because like tend to the people's face, another name is called the kissing box. Can drink up 300 milligrams of blood in 15 minutes. That's a lot. And if you see the picture, this is after suction blood meal. It's really huge. But what is chemistry behind it? So this is a look like the suction bug. And this bug, the bug is very smart. So, but the bug may carry some of the parasite and the cruise. This is the vector of the Chagas disease. This disease can be very dangerous. It's a non-curable. It's you can find in South America, so they're causing the 43,000 died annually, so that's quite a lot of number. And then you cannot find a cure when it's getting long term. So basically damage your brain and damage your heart, so it's very bad. The protozoan is passed through the feces, amphibiatic feces, which the insect leaves behind in the vicinity of the wound. If you don't scratch it, it's okay. But when you scratch it, you push the feces down to the blood, and then that causes trouble. And what is chemistry? So it turns out the saliva of the bug contains a heme angle. When you suck it, poke a hole on your face, on hand, they will give the heme angle to your blood. The angle will release it and make your the muscles relax, so they can suck more blood. But for your immune system, when they have the tissue trauma, the immune system will form the histamine. And histamine will make you swelling and itching, so you will notice that. But this bug is very smart because they also give the heme. So the heme combined with the histamine, what's happening is the heme angle, the angle relax your muscle, the nitroglycerin, so that is a heme protein. So they bind with the histamine, so you won't feel the itch. Until the bug leaves, you start feel because there is no heme angle anymore. So you start feel itching and swelling. So it's very smart. So this nitroglycerin is heme protein also. and after releasing enol bond with histamine, and thus preventing inflammation and itching during the feeding. So basically, you won't realize until they leave. So very smart. So the kind of chemistry is neat and relaxed. So that will avoid detection by the victim. We talk about a lot of examples. They're all talking about heme and enol. And so this is a kind of, I just show again the heme, the structure, what it looks like, and heme structures. And you can find it in the heme enol synthase, cytochrome P450, and myoglobin, hemoglobin. And so that is very important. And I talk more now, from now it's more like the research, what I'm doing now. So I give the title is heme, nitrite, enol, because we know the enol can convert it to the nitrite. Nitrite also convert it to enol. What reaction when it's react with heme? The nitrite is common in the universe. You can find it in the water and in many places. And it is used for curing the meat. So if you go to the grocery store, normally you will buy the meat. You will choose the color, kind of look fresh. But look fresh is not means fresh. Okay, what people done for treatment, you will see the color getting oxidized myoglobin, basically give you the brown color. It's kind of old, you don't want to buy. But when they treat it with nitrite and cheated on you to give you the myoglobin enol color, but similar like the oxygen, it's kind of very fresh. But you cannot cheat by the colors. So this is called cured meat, and also a way to kind of make the ham and the bacon. They use a nitrite cured meat. So it's very kind of, we saw it in the daily life. Okay, so now it's if you remember when you form the enol in the biological system, you need oxygen. On some circumstances, oxygen may not be that much for the enol produced. So it's another alternative way to make the enol in the biological system, especially in the hypoxic, just like a less oxygen concentration. The body still can produce enol where it's formed. So it has been proposed to study and proposed the nitrite can form enol under hypoxic conditions, under lower oxygen concentration conditions. So it can be treated as enol donor. So this is a denitrification process. Basically, the natural to form if you find it in soil, mostly it's a nitrate, and then some bacteria to reduce it to the nitrite. And nitrite use a nitrite reductase enzyme. The heme also is a heme-containing enzyme to reduce nitrite to enol. And then continue, eventually form the nitrogens, and then go back to the atmosphere. So this is a process called denitrification. Basically, it uses the inorganic nitrate back to the nitrogen and to fulfill the nitrogen cycle. So if you go to nitrogen and go to ammonium, so that's a fixation, nitrogen fixation. Basically, you use nitrogen to ammonium. So this is it's kind of like a nitrogen cycle so involved. And the one step is heme ion mediating the nitrite redox activity. So basically, it's the porphyrin, the heme porphyrin contains can react with nitrite and present the electrons, and you form the enol and water. And this is a enol combined with a heme protein, and so that was the meat color and showing the fresh color. So that is one of the reactions. If you, I don't know whether you guys already teach the nitrite. Basically, it's just 3 atoms. Nitrite ion is 3 atoms, 2 oxygens and nitrogen. If you think how to bond over the heme, you would think about this is an ion, how to bond it. Because what we're saying is, what is the reaction going on? It starts with the first stage. If the nitrite bond up to the protein, it determines the pathways next reactions come on. So it's very important to know how the nitrite bonds to the protein. So if we know, this is the 2 major ways you find out the nitrite bond protein, either bonding through the nitrogen or bonding through the oxygen. We are interested in the bonding modes because that is carried out the next stage. So that is determine the mechanism of the pathways, the next reactions going on. So that is very interesting for us to study that. So the N-nitro mode is most common for iron heme model compounds. Basically, you will see this is a major bonding mode, and this is an uncommon bonding mode, and normally found in the mammals. So, as I said, bonding mode determines the reactions, and another thing is the energy difference. If you use any bonding mode, that is energy is lower, it's more like a stable, because energy lower is more stable. So it's around 4 kcal per mole. What does that mean, 4 kcal per mole, the numbers? Is the hydrogen bond in the biological system, the energy, if you break up the hydrogen bond, water bond, the energy is around 3.6 kcal per mole. So that means if you want to switch from the N bonding mode to O bonding mode, you don't really need that high energy to do that, because hydrogen bond energy will give that energy to switch. And this is maybe it's kind of new to you guys. This is protein, the active site protein structures. And we use the blue to stand for nitrogen, red for the oxygen, and the gray one for the carbon. And the middle one is the iron, it's brown color, but does not show that quite well. So this is the active site, and if you remember the heme, the structure there, so if you colorize it, it's more like this one. This is more protein. And that porphyrin is not flat, you can bend a little bit. What I pointed out is prior to our group studies, you will see most of all the bonding mode and see the N bonding mode. It's a stabilized bonding mode. And this is carried out nitride reductase activity. But our group, its first group get the first structure is showing the O bonding mode. And this is in the myoglobin, is hostile, is in mammals. And the previous one is in bacteria and fungi. So it's different system. It's a mammal system and versus the bacteria and the fungi. So you will see now, before our structure, you're thinking about for the bacteria, for the mod compound, all show the N bonding. Without some surprise, you should also get the N bonding for the mammal, for the myoglobin. But it turns out, no. So the mammal protein, the myoglobin, is very special to forces O bonding. Even we know this energy will be higher than the N bonding mode, but it happens. And what we did next one is, this is natural, the white type protein. So this is natural from the muscle. If we do some mutants, what we mutants, we change something on the protein. We kind of not use the natural heme protein iron that is contains iron. We replace it to manganese. So it's one on the left, on the periodic table is left on the iron. What's happening is it still maintains O bonding mode. So the protein is stay forces O bonding mode. And we also did another work is what happens, we replace iron porphyrin to cobalt. On the, in the periodic table, cobalt at the right side of iron, so it's 1 electron more than iron. So you still get the getting the O bonding mode. It's like, so no matter you change the oxidation state, and you would think about the cobalt 3 is more like iron 2. Maybe, I don't know whether you guys cover it or not. So it's because iron is a D5 orbital, it's iron 3, and the cobalt is D6, is 1 electron more. So the cobalt 3 is iso electron like iron 2. So basically, and the special one for this one is all the cobalt model compounds, nitride complex is all showed N bonding mode. So when it goes to protein, you force it O bonding. So that protein plays a role to determine, to direct the bonding modes of nitride. And I show you it's more like the active site. This is a protein like a ribbon style of the protein structure. So the myoglobin is 8 helices. So this is a folding rod, it's more like a neat monomer. So the heme side is kind of close to the surface, exposed to the water. The solvent is here, and half is hiding in the pocket. So we use we call the pocket bonding to the ligand, called as a distal pocket. The bottom one we call the proximal bonding site. And this one is a little bit more complicated. So this is a hemoglobin structure. So it's a tetramer. So it's like a 4 heme site. Instead of monomer, it's just a 1 heme site because the tetramer is an alpha 2, beta 2 is a heterodimer. So this is a protein structure. And this is folding, but it did not show the side chain. So we know the myoglobin nitride is O-bonding, and no matter you change to manganese or cobalt, it's O-bonding. But how about the hemoglobin? Because hemoglobin, you think about red blood cell, 9% of the weight is hemoglobin. So it's very, very concentrated and very important. If you if you add nitride to the blood, to your red blood, you will see the color of the blood turns to brown color very quickly to oxides and form the methemoglobin. And the methemoglobin, in normal, everybody body contains methemoglobin, 1 to 2% of the methemoglobin in your body. But when you get it higher, like 20%, you get a little bit offended, 50% you become nauseous and bad, and 70% you're dying. So, but you can treat it, the nitride, the methemoglobin with a reducing agent, so like a methylene blue. So you have you called the blue baby syndrome? The newborn baby, and they show the blue color, but they show blue color because they have a higher concentration of the methemoglobin. So what's happening is brown color of the hemoglobin on the skin should give you the purple color, blue color. So basically that is because maybe though from the drinking water, the feed contains a high concentration of nitride, so that is causing their methemoglobinemia. So there is a name called methemoglobinemia. So, and how nitride binding to the hemoglobin? We know binding to myoglobin is O-bonding, but how is the hemoglobin complicated one? So this is the active side of the methemoglobin structure. You will see the methemoglobin, the pocket binding with water. So this one, red one, the dots look like it's water. We stand for oxygen. You won't see the hydrogen in the protein structure because it's too small. So you just see the oxygen. And when you react with the nitride, it's also showing it's O-bonding, but a slightly different bonding mode. So you will see this one will similar like myoglobin is O-bonding in this format, but in the beta, in the different subunit, it turns out a different bonding mode. So this is kind of neat. And this is a crystal, protein crystal. So yesterday, Jason and I, we went to Fletcher to get some thallium crystals. This is a crystal of a protein. Yes, so if you say this is a crystal of the blood hemoglobin, so it's a red. After I tried it, you see that this one have two forms. Maybe this is not that clear, but they the big chunk diamond shape, the crystal, and this is a solid structure like give you this. And they also have a needle shape, but it's too small to get structure out. Basically, you can get crystal of the protein and solve the structure to see what the protein looks like. So that is what we're doing. So this is repeated. So you will see in the alpha subunit, you get O-bonding, but in the beta, you get the transformation, the cis form. But we were saying because if your bonding mode is slightly, conformation slightly different, that also determines the different level of the energy. So, and they have a reaction will be different. Okay, we do, maybe you haven't taken the biochemistry class, so you may not know his 6,4 stands for what. His stands for histidine, it's an amino acid. So the 6,4 means the number, the sequence, the location of histidine residue in the protein. Basically, it's if you go to the myoglobin sequence, because it's 8 helices, the number 64 residues is histidine, so that is what we name it. Use his 64, the residues, and direct bonding, because what I was saying in the pocket, they have the histidine can forming a hydrogen bond to bond with the oxygen of the nitride. So this residue is very important to direct the bonding mode of nitride. And we do some mutagenesis, is what I would say, we mutate the histidine 6,4 to valine. And we know the valine cannot form the hydrogen bond to the nitride. Basically, you remove it, let it free. And we also do is introduce another hydrogen bond at different location. What will happen? So this is structure you already seen in the previous slides is O-bonding. So if you do the mutagenesis, you remove the histidine, and before you get the metaphone, you see the pocket is empty. But the metaphone of the white type histidine 6,4 protein, you will see water in the pocket. But in the mutant structure, you won't see anything in the pocket. It kind of distal pocket is empty. The green one, the measured one, is kind of a density to determine what's in that density. So this is you will see the model is perfectly fit with the density it's showing. There's no density in the pocket. But when you introduce the nitride, so now you will see the density. We'll model it. So what do we see? This is the density you see. You will try to put a molecule in that density to see how it fits. And now it's more like the in-bonding mode. And what tells us is, if you move the hydrogen bond in the pocket, and you let the nitride find the best conformation for the protein, you will get the in-bonding mode. That is consistent to the model compound without all the amino acid around it. You get the in-bonding, so that is stable mode. So this one is after you move the density, we'll see this is a model. It's how do you put it, the protein like bond and the heme part. You will see the no water in the pocket before. After you add the nitride bonding through the N mode and the water in the pocket. So this is kind of unique. You will know the histidine is important. And then later on, what we do later is we introduce another hydrogen bond residue. In this case, it's arginine, because when you take the biochemistry, you will know the arginine has a long side chain and have more freedom to frame in and frame out. So this is a kind of a structure of the arginine. You will see in the density a very long side chain and pointing to the out of the protein. And there's nothing water in the pocket. But when you introduce the nitride, amazingly, this is happening in the crystal structure. You will see the crystal is like a plate. It's very rigid. And what happened to the protein is this arginine can swim into the pocket and bond with the nitride and introduce another water and force this propionate group to facing and down. And that is a lot of movement we're taking. So this is the protein, the model. We will model to the two proteins. This one is made for without the water in the pocket. After you add nitride, the side chain of the arginine swimming towards the nitride and force the propionate group to face it down. And I show you. So this is the overlay of the two protein structures. You will say that involves some side chain movement. This is zooming the active site, and you will see when the nitride comes in, the side chain of the arginine swing starts moving, but you will see the backbone of the protein does not move much. It's just the side chain of the arginine and the propionate group of the heme and one of the residue at the bottom. If you see this, the backbone is kind of rigid. The movement is involved the active side. And what this tells us, you will see this is white type, you force it O bond. When you move the hydrogen donor, the histidine, native free antibody, you will get it more like an inbound smaller compound to the other bacterial protein and fungal protein. But when you introduce another residue in the distal pocket, it's kind of a force at the O bonding again. So these tell us the histidine 6,4 direct the nitrite as O bonding to the protein. So makes the mammalian protein unique, because you see the end bonding is in bacteria and fungal protein, but a mammal is myoglobin, hemoglobin, because the one histidine in the distal pocket and forcing direct the O bonding. And of course, I would like to give applause to my supervisor, Dr. George Richardo. He's the chair of the department. And then Thomas, he's the one for the protein crystallographer, the manager, who helped me to solve the structure. And Peter Ford and Julie Hennekins in their UC Santa Barbara to the kinetic study to study the nitrite reductase of the mutant protein myoglobin, how fast it is. Because we know the myoglobin, the purpose of myoglobin is to storage oxygen. It's not for the nitrite reductase. Later on, we found out when the human body in the biological system, when the oxygen level is lower, the body can still produce in what platform. So people studied hemoglobin, myoglobin, found out hemoglobin, myoglobin can have the nitrite reductase activity. So when you start for the active, the white type myoglobin, you can see it's faster than mutant protein. Glenn Morris and he's in the OU Health Science Center. We use his X-ray instrument for the structures. This is the funding from the OCAST and NIH. So this is a new building, our new chemistry building. So this is, as I said, it cost $80 million. And the 3 stages to hold 30 PIs in the building. And if you go inside, this is a kind of like, maybe it's a little bit dark. So we have called it the open lab. So basically, on each of the wings is hold like 5 PIs, 5 principal investigators. You will see there is no doors between the labs. You can from here you to the end of the hallway. So basically, all the labs in each wing are connected to each other. So if you have questions, you can ask. You're very neat. So you can design at the beginning. You can design how the lab you want it to be. So basically, our lab is on this side, but it doesn't show. So this is Dr. West's lab. She's on Vikings. So she's doing the protein crystallography also. So this one is a student's area. So one of the examples. So we separate the lab with the student's area. So basically, the student lab, you cannot bring the gloves and chemicals in the lab. That is just for reading and eating and place. So it's very good, but they have a windows here, so basically you can see the lab through the student's area. If you're running some experiments, you can see it monitor in the studio. You don't need to sit on the bench. This is a hallway on the 6th floor, is outside of our labs. So this is kind of faculty offices. And a unique part of the buildings, they have a lot of conference rooms, small one and big one. So if you want to meet with others and have a discussion, have your kind of like office hours, you can, don't disturb in the students area, you can find a small conference room, you can sit down discussion. And this is kind of again, one is, this is stairs, and we have you will see this is not finished yet, still need to put the pavement and still need to put the grass here. This is courtyard, and this is the stairs. You'll see they have kind of here benches, and you can lie on here and study here. So you can see what's happening in the courtyard. It's very nice. And this is the first floor. They have a small cafeteria. It's like called Element Cafe. So they have both, of course, cafe and drinks. And they also sell some simple non-drinks like a sandwich and salad. And you can eat here. So they also have some couches and desks, so you can take a nap and reading there. So this is the lounge at the 2nd floor. So basically I have 2 make couches and have some comfortable chairs so you can reading and meet with a friend there. So you can, it's very nice. And that's all. Any questions? No. About chemistry, about, okay.
I was just wondering, you had nitroprusside and you had nitroglycerin.
Mhm.
Is nitroprusside, is that the less explosive one? Is that the one that they would use for like if someone has angina or?
Yeah, angina bronchitis, they use it's called nitroglycerin. What is happening is put it on the tongues and then give you, yeah, so nitroprusside is less toxic, they also, yes.
Okay.
So how long does it take to make a protein crystal?
It depends. It depends. Sometimes it takes 1 year to. Okay, the project that was take a long time. When you get the right conditions for myoglobin, hemoglobin, mostly you grow the crystal within a week. You will see the crystal, and on the microscope you can also see some growing every day, day by day. So you just choose the right size. A little bit bigger because you also determine the size. If too small, fewer data spots, you can solve the structure, so less point for the structure solutions. So you need a kind of suitable size. Normally it's 0.1 millimeter, 0.1 millimeter, and 0.1 millimeter. So that is a kind of good size. You don't want too big, you don't want too small.
So what was the longest it took you to grow a suitable crystal?
One of the projects took me 1 month to show the crystal, and I give it up and I try to throw the plate and find out the crystal after 1 month. Because the thing is like sometimes you set up crystals, you watch and tomorrow, day after, and after 1 week it's that lose the hope, is maybe nothing happened. And one of the crystals is heavily precipitate. You will see the precipitator from drop. You didn't see anything. After 2 weeks, I kind of maybe this won't work. And after 1 month, and then someday I'll try to throw away the trace and find out the crystal. Oh, wow, good. So that is took 1 month. So the longest one yet. It's good because for the heme proteins, good side is colored one. It gives you pretty color. The myoglobin is more like a brown color, but the hemoglobin get a red color. It's very shiny, very good. It's, on a microscope, it's like a ruby. It's very nice crystal. Yes, so that is a good side to work with heme protein.
So what future directions do you see this going?
Oh, we found that, like I said, what is the nitrite? Nitrite is good because formed in a vasodilator. It's relaxed and also it's good. The bad things about nitrite, it causes the methemoglobinemia. Right? So because oxides the hemoglobin give you bone cancer. Because why the methemoglobin is toxic? Because the methemoglobin cannot bond with oxygen. The purpose of the hemoglobin is transport the oxygen in your body. When it's formed the methemoglobin, now even you have oxygen around, the hemoglobin won't bond it. So that is the lose the function. So of course you can reduce it back to deoxyferrous hemoglobin and then rebond with oxygen. The ugly things about nitrite, even put a high dose on it, it will cause the Hens body. Hens body is kind of a fragment, agree together, attach on the membrane inside the red blood cell, eventually damage the red blood cell. So that is irreversible because methemoglobin is reversible. You can add reducing agent to reduce it back, bond with oxygen. But when you have high concentration of nitrite, it causing you damage the red blood cell. So that is ugly part. But how that's happened? And you will study for the hemoglobin with nitrite, you will see actually what's happening is the heme is kind of slightly moved out from the protein. It's kind of getting then aggregated and then causing the damage of the protein. So it's kind of unique in what the next step. So we'll be going, and of course, it's a study the nitrite reductase activity of hemoglobin. and what is self-causing.
Did you get this idea to begin with? Like, how did you come up with this track of research?
Yeah, actually, what I can tell you the truth, at the beginning I would just say, okay, we get myoglobin nitrite, it's so funny. Maybe it's cool to get a hemoglobin, because hemoglobin is the tetramer is more complicated than myoglobin. We just need to get a structure, what it looks like. And then when you do the reaction, you will see even the simple reaction, add nitrite to hemoglobin, sometimes give you different colors. So the red protein, when you with the nitrite, eventually get the green color. Why does this happen? Yes, it's because blood is red. When you add nitrite, you kind of get the things is kind of green. And then later on we get the different structures, and we'll find out actually some more reactions going on even on the crystal structure. And the same thing, I'm talking about the curing meat. You add nitrite to the myoglobin, to the meat, right? Sometimes you will see that if you pay attention, you see the edge of the meat is showing the green colors. That is not malt. That is not. It's some reactions going on because of nitrite with the myoglobin. That is neat. What is that green color pigment? They call it the nitrite burning, and or it's called the nitroheme. It's showing the green color. So that is a nitrite chemically and to modify the hemoglobin. It's showing the green color. The meat, of course, you don't want to eat that meat. It's already some chemical reactions coming on, and but it's chemistry. If you get the base, it's like a small molecule reacted with protein, gives some interesting results. So it's very exciting for me. I don't know for you guys, but it's very kind of related to the daily life, and then you can find experience. If you see the meat on the edge is green, definitely you won't think the first thing to think about it, maybe it's mold, it's something bacterial. It's not. It's just a nitrite. It plays the tricks.
What kind of software do you use for those X-ray structure?
Oh, we have for the protein crystallography, we have so they have kind of open source softwares, and you can download from the website, CSP4, they can come, yeah, so it's kind of free. They have a whole bunch of the software, but they have several stages. So for the protein structure, from the crystal you were showing the diamond shape to the final structure, it's not one jump to the end. You have many steps. First, you use X-ray to expose on the crystal. You will see the spot of the diffraction patterns, and then you try to decode that patterns to some of their structural factors. What is the intensity of the spot, intensity of the spot, and then try to find a solution, and try to find the phase problem, and then try to solve the structure. So it's kind of the 5 stages. It's not just one to the end. So sometimes it takes a long time. Students? Oh, you can also ask me about OU, chemistry. I cannot tell you much about OU, but chemistry department, maybe I can ask some questions.
Normally when there's no noise, they really have questions. You just have to stare at them longer.
It's okay. So you can email me.
So are there any other questions from anybody? So don't let us monopolize all of your questions. Okay. Well, then let's give Eva a hand for